Good morning, everyone. Thank you all for joining me today. My name is Navya, and I'm a PhD researcher at RMIT University, Melbourne. Today, I'll be presenting my research titled Biogenic Carbon Dots derived from the microwave carbonization of amino acid mixture. This work explores the synthesis of biocompatible carbon nanodots from amino acids using a simple microwave assisted method, with applications in bioimaging and sensing. In biomedical applications like imaging and detection, the choice of nanomaterials is critical. We need materials that can visualize cells and tissues and detect analytes with high sensitivity. But the challenge is that many conventional materials fall short. Traditionally, organic dyes and quantum dots have been used. Organic dyes are easy to use, but they come with major limitations. They tend to photobleach, have low signal intensity, they accumulate non-specifically in both healthy and diseased cells, making it hard to get a clear contrast. Quantum dots, on the other hand, offer strong and tunable fluorescence and good stability, but they contain heavy metals like cadmium or lead. This raises safety concerns, especially for in vitro and in vivo studies. Complex surface modifications are often needed to make them biocompatible, which adds to the cost and complexity. There are also environmental concerns with their disposal. And when it comes to detection, traditional methods are often time consuming and expensive. They usually require external labels or contrast agents, which can complicate the process. These factors limit their use in point of care diagnostics. That's why there's a need for new nanomaterials that are safe, effective, and functionally adaptable, especially ones that can address the limitations in imaging and detection. Carbon dots have emerged as a promising nanomaterial for bioimaging and detection due to their excellent properties like fluorescence emission, good photostability, high water solubility, good biocompatibility, small size, and ease in surface functionalization. In our study, we are particularly focusing on using amino acids as precursors, and there are several reasons for this choice. First, amino acids are naturally occurring, biodegradable, and water soluble, which eliminates the need for toxic solvents and harsh chemicals. So, from a sustainability perspective, this makes the process eco friendly and biocompatible from the start. Secondly, amino acids are chemically rich. They contain functional groups such as carboxyls, amines, and side chain elements like nitrogen and sulfur. These heteroatoms gets incorporated into the carbon core during the synthesis process, which allows us to achieve heteroatom doping and surface functionality in a single step. And the third reason is the choice of amino acids gives us the control over the properties of carbon dots. So different amino acids leads to carbon dots with tunable fluorescence and dispersibility. Carbon dots can be synthesized using top-down or bottom-up approaches. Generally, bottom-up methods like hydrothermal and microwave treatments are most popular for biological applications. Precursors like citric acid, glucose, organic waste, and other organic precursors determine the size, emission, and surface groups of the resulting carbon dots. Though there are a number of different organic precursors that have been used to synthesize carbon dots, many of these suffer from poor quantum yield and require complex surface modifications to make them useful for biological applications. Here in our study, we are typically using amino acids like glutamic acid and tryptophan and their combination. Glutamic acid contributes abundant polar surface functional groups while tryptophan introduces nitrogen doping, enhancing the optical and electronic properties of these carbon dots. So when used together, the mixture of glutamic acid and tryptophan offers a synergistic effect, combining surface functionality with enhanced optical properties. So herein we have synthesized carbon dots, three different types of carbon dots with individual amino acids as well as their combination via a microwave assisted approach. The optical properties of these carbon dots were studied using UV visible and fluorescent spectroscopy. An absorbance peak at 275 nanometer of tryptophan carbon dots and glutamic acid and tryptophan carbon dots is due to the N-pi star transition that contributes to the photoluminescent signals. However, the N-pi star transition in glutamic acid and tryptophan carbon dots is much broader compared to tryptophan carbon dots and may be due to the formation of numerous surface defect sites. It also displayed an additional absorption band centered at around 350 nanometer, which is probably due to the formation of new electronic states. And if we see the fluorescent spectra, the glutamic acid and tryptophan carbon dots exhibited intense emission bands centered at around 450 nanometer, while the tryptophan carbon dot showed moderate emission. Conversely, glutamic acid carbon dots demonstrated lowered emissions when compared to both other two carbon dots. So with this, we can infer that carbon dot formation occurs preferentially from tryptophan rather than glutamic acid. However, the carbon dots that were formed from the mixture of tryptophan and glutamic acid exhibited 2 times stronger emission as compared to carbon dots synthesized using pure tryptophan, even though the concentration of tryptophan to form glutamic acid and tryptophan was half the concentration that was used to synthesize the tryptophan carbon dots. This indicates that glutamic acid was found to play a major role in enhancing the photoluminescence of these glutamic acid and tryptophan carbon dots. The fluorescence characteristics of these carbon dots depend on the extent of carbonization, which is influenced by the reaction conditions. So the reaction conditions were optimized to enhance the fluorescence properties of these carbon dots. And here we have changed the microwave power, reaction time, and glutamic acid and tryptophan ratio to see what reaction condition will yield us highest fluorescence. And we could see that when the microwave power was maintained at 200 watt and after 120 minutes, and when the ratio was equal, we could see the maximum emission at 450 nanometer. However, the emission maxima did not change when did not change the position when the ratio of amino acids were varied. So these results demonstrate that the fluorescence emissions can be adjusted by varying the ratio of amino acids, microwave power, and time. And the optimized production of these strongly emitting carbon dots is likely attributable to the formation of graphene structures derived from the aromatic group containing tryptophan and surface functional groups contributed by glutamic acid. The size of the carbon dots was measured using high-resolution transmission electron microscopy, and we could see that the carbon dots displayed spherical morphology with an average size of 6 nanometer. The interplanar spacing distance of 0.28 nanometer indicated the graphitic core structure of glutamic acid and tryptophan carbon dots. Further, we went on analyzing the surface functional groups of these carbon dots and the X-ray diffraction patterns of these carbon dots. As we can see here, the glutamic acid carbon dots displayed a broad OH stretching, which was absent in the glutamic acid tryptophan and tryptophan carbon dots. This also indicates that glutamic acid did not chemically transform into carbon dots. And the carboxylic peak was also absent in the case of glutamic acid tryptophan and tryptophan carbon dots. And to understand the structure, X-ray diffraction patterns of the carbon dots was collected, and glutamic acid carbon dots exhibited an intense crystalline peak, which again supports that glutamic acid didn't chemically transform into carbon dots. The sharp multiple intense peaks suggest the ordered crystalline structure, which leads to fewer defect sites and lower fluorescence. In the case of tryptophan carbon dots, they displayed broad and low intensity peaks, which is typical of amorphous carbon. Bulky indole ring disrupts the graphitic stacking, leading to more disordered structure, enhancing the photoluminescence. And the XID pattern displayed by glutamic acid and tryptophan carbon dots showed a weak and broad diffraction peak centered at 23.52 theta value, which corresponds to 002 lattice plane. indicating the formation of a graphitic-like structure. And this results agrees with the calculated interplanar spacing from high-resolution TEM image. So with this analysis, we could say that glutamic acid and tryptophan carbon dots possess polar functional surface groups and they exhibit a graphitic core structure. To understand the chemical composition of the carbon dots, X-ray photoemission spectroscopy analysis was carried out and is shown here. As we can see here in the survey spectrum, it displayed oxygen, carbon, and nitrogen only. The C1s in the level analysis showed that there are 4 different kinds of carbon present in carbon dots, and these 4 peaks corresponds to saturated, graphitic carbon, carbon nitrogen, and carbonyl groups. The N1s analysis showed that there are 2 kinds of nitrogen corresponding to graphitic nitrogen and the protonated nitrogen. And the O1s level analysis showed 2 kinds of oxygen, probably originating from the surface hydroxyl oxygen groups, and carbonyl oxygen, which is in agreement with the FTI results. So the XPS analysis confirmed the effective nitrogen doping and presence of polar surface functional groups. The inherent fluorescent property of these as prepared glutamic acid and tryptophan carbon dots was used for the quantitative detection of cysteamine. As we can see here, there was no shift in the emission wavelength observed, but the emission intensity was found to decrease with the increasing cysteamine concentration. And the limit of detection and limit of quantification were found to be 1.35 micromolar and 4.5 micromolar. Further, to validate the selectivity and specificity of glutamic acid and tryptophan carbon dots for the detection of cysteamine, an interference study was conducted. Here we have used different interfering cations and intracellular thiol molecules. As we can see here, only cysteamine could induce obvious quenching towards glutamic acid and tryptophan carbon dots when compared to other interfering substances. So these results demonstrated that the glutamic acid and tryptophan carbon dots exhibit very high selectivity towards cysteamine. We further studied the cytocompatibility of these materials and the influence of carbon dots on MCF7 human breast cancer cell and fibroblast cell toxicity was studied using MTTSA. And the cytotoxicity measurements were made with different concentrations of carbon dots ranging from 0 to 50 microgram per ml. The average cell viability remained above 80% for the highest tested concentration, demonstrating the biocompatible nature of these carbon dots. This is the comparative fluorescence images of untreated and carbon dot treated MCF7 cells. The imaging results demonstrated a significant contrast between the treated and untreated samples. While the untreated cells showed negligible fluorescence, those incubated with glutamic acid and tryptophan carbon dots displayed strong clean fluorescence, indicating their efficient uptake. The strong and stable fluorescence, along with the demonstrated biocompatibility, positions these amino acid derived carbon dots as promising candidates for bioimaging applications. So to conclude, carbonization of tryptophan and glutamic acid contributed to the formation of graphitic carbon core and surface polar functional groups, carbon dots. And the combination of amino acids influenced the photoluminescence behavior and dispersibility of these carbon dots. And these carbon dots were stable across various pH and salt concentration, and photostability was observed. These carbon dots also demonstrated biocompatibility and selectively detect cysteamine and has the potential to be utilized as bioimaging agents. Finally, I would like to thank my supervisors, Distinguished Professor Suresh Bhargava, Distinguished Professor Magdalena Plebansky, and Dr. Ruchika Oja. Now, also I would like to thank members of CAMIC and KAVA Lab and the PhD scholarship from Australia India Research Fund, facility at RMMF and MNRF, technical staff, and University of Miyazaki, Japan. Thanks for your time and attention, and I'm happy to take any questions.